ठीक है ठीक है ठीक है.
Can you see the pointer, Dr. Pawar?
Yes, sir.
Okay, perfect. So dendrimers are basically the fourth class of polymers. When I say polymer means they were linear polymer first, like nylon and all those things which we use. Then came branched polymer. Then came hyperbranched polymer, and then the dendrimer is the fourth class of polymer, which is spherical in shape. It has three main parts. It has a core, and from the core we develop branches like a tree branches, and then you keep on adding each layer like an onion. You keep on adding each layer. So this is generation 1, then next generation, generation 2, generation 3, and keep on adding. At certain point it all ends like this. It's like a very old mango tree. It's properly, you know, so many mangoes are there. It's totally closed basically. What it does is that you can entrap the drug in these spaces here, right? So you can do the molecular entrapment. That entrapment could be a drug or any other molecule. Also, you can chemically conjugate so many things. So, and you will say why it is important. Because many of you who work in the area of drug delivery, so you either use some kind of a micelles or liposome or nanoparticles or cyclodextrin or emulsions for your work, right? So each system has their own advantages. But any of these systems like micelles or liposomes, even nanoparticles, doing chemistry over surface is not easy. It's not easy to chemically modify a liposome. It's like a very sticky thing. It's like if you have a glue in your hand, it's difficult to put something else somewhere else, right? So because of dendrimer, it's a chemical structure. Doing chemistry is easy. Means you can modify it for your requirement very easily. So the two main advantages of dendrimer is having a three-dimensional structure, precise size, and ability to modify chemically basically. So dendrimer, which I work, is called PAMAM, polyamidamine dendrimer, was developed by Dr. Donald Tamalia, who is now my collaborator also. I worked under him and now I work with him as a collaborator many years back. And at the time, people were saying that when the polymer was at full swing, that you make polymer, you take monomers and throw UV light on that. And then it forms polymer, like a bunch, right? But they don't have specific sizes. A polymer could be from 10 Dalton to 10 million Dalton within one population. So he came with the idea of making a uniform polymers, which are dendrimers. So from that time, this field has grown a lot. If you see here, I was talking about three types of polymers, like a linear polymer. Then they made like a cross-linked polymer. Then they made branched polymer. Then came dendritic polymer. Of that, dendrimers are one of the four. So Tomalia and a few other made dendrimer. What I have done is, and also what they thought, because dendrimers, depending upon their surface, there are different kinds of dendrimer. If surface has amines, they call amine dendrimers. If surface means outside here, surface outside. Surface have carboxylate, they are called carboxylate dendrimer. If it is hydroxy, they are called hydroxy dendrimer. If it is a PEG, polyethylene glycol, it's called PEG dendrimers. And each dendrimer behave differently, and I have proved it through my publications and patents. So they also, what they thought, what if take two different kinds of dendrimer and chemically conjugate it? So they made something called megamers, sticking two balls and attach it, right? But then they have to do it chemically. They have to do chemical reaction to make something, and these are called megamers. So what I have done here is that I came with a new idea. I said, I'm not a chemist. I don't like doing chemistry. I have taken two dendrimers and basically clicked them without any chemistry. No chemistry involved. A simple technique, which I have a patent on, where I can take two dendrimers, different kinds of dendrimer, and click them together. And I name them as a hybrid dendrimer. Like two balls, I'm clicking together without doing any chemistry. So now my work is on that, how to use those two dendrimers and utilize it for drug delivery. So that would I will explain what I am done. So this is an architectural view of hybrid dendrimers. And you have seen here that I have taken one dendrimer called amine dendrimer and one called carboxylate dendrimer. And I clicked them, and you see how they form an architecture. So you can see green and brown dendrimers is stick together. And with that, I can entrap drug. And in this research, we have used docetaxel, an anti-cancer drug. So my idea is that I can do a lot of chemical modification in each one of them and create a nano device. What do you mean by nano device? When anything goes inside the body, you don't have a control. When drug is inside the body, you lose. What should we do that we can have a control? It's like a robot going inside the body and listening to you. Can we do that? Right? Think about that. Almost 130 years back, did we think about a car? We had some bullet tracks and we have a concept of wheel, right? But we thought of wheel and we were using horses or bulls to carry those wheels, right? But then with the invention of gas and electricity later on, the idea of force, this whole thing brought us to cars basically, right? Similarly, and that car needs a chassis and a motor, depending upon what you're using, electric or gas, different things you need. So you need a structure to develop basically. That's what we're trying to do. Trying to create a small robot where we can modify it like a drones, right? You might heard about drones nowadays, that drones goes to any place you want and control from outside. Something like that we are trying to create. So this is kind of attempt towards that side, but inside the body. So glioblastoma, brain tumor, I told you already, difficult to treat. People don't live after recurrence long. Temozolomide is one drug for choice. People use it. But here in this research, we use docetaxel. The reason is that this docetaxel has solubility issues of their own, and it cannot cross blood-brain barrier. And here, my idea is to show that this hybrid dendrimer can help to cross blood-brain barrier. So pick docetaxel. This comes in the market as a Taxotere, is its brand name. So we are trying to achieve two things using this hybrid dendrimer. One was, can we increase the oral delivery? Means when you take something orally, it goes to GI tract. From that, it absorbs and permeates and goes into the blood. That's called oral bioavailability. If drug has low bioavailability, less drug will reach blood, so less chance of reaching your target area. So one thing, because this does have low solubility, can we increase its bioavailability? That's one objective. Second was, can we create a multifunctional nano device using multiple targeting ligands? But that would be IV injection. So I'll explain you this as we move on, each objective. So objective one, to develop an oral delivery of docetaxel. So you can see here that what we have done, because we have used amine dendrimer and carboxylate two dendrimers here. So first thing is that we will take single dendrimer and entrap the drug, docetaxel or DTX, single, and then we prepare the hybrid dendrimer and entrap the drug. And then we studied all the parameters. You can see this is a transmission electron microscopy of dendrimers. You can see this is how dendrimer looks, A. This is dendrimer after loading the drug. You can see the size is slightly increased. Here is dendrimer second, which is carboxylate dendrimer. And after drug loading, looks like this. And you can see that here, this is your hybrid dendrimer, different kinds of. And you can see that here, This is hybrid dendrimer, and you can see the size is bigger compared to single dendrimer. So it shows that that thing is forming, right? We can see visually by microscopy. And when you load the drug, you can see the size has further increased. So we have proved by TEM that this dendrimer forms as we thought of it. Then we did the NMR and all to confirm whether drug is entrapped in the dendrimer. Similarly, we did the stability study, means if we keep it for some time, like for 6 months, are they stable by zeta potential? And we did the hemolytic study about the further toxicity. If you incubate this formulation with RBC, what will they break RBC or not? You can see that compared to Taxotere, which is 11% hemolysis, these formulations were very safe. Similarly for stability also, it's more safer and more stable. So you have to remember here 3 things, what we are testing. We're testing drug alone, this Taxotere, single dendrimer, A means amine dendrimer, C means carboxylic dendrimer, and then we are doing hybrid dendrimer, mixing A and C together. And you can see that drug solubility increases with increase in concentration in both dendrimers. And you see that it is more with the hybrid dendrimer here. Solubility increased very much, almost from 6.8 to 100 microgram per ml. Similarly, if you see the release of the drug in water and the gastric medium, you can see that compared to Taxotere, which is a market drug, all dendrimer formulation showed faster dissolution, not release, faster dissolution, both in water and the gastric medium. Why it is important? Because until and unless when you take a tablet or capsule, it dissolves in GI medium. GI means there are 2 GI mediums, gastric medium and intestinal medium. And remember, both have separate pH. Gastric is pH 2 and intestine has pH, basic pH basically. So drug must dissolve. And solubility has many issues. Solubility of a drug depends upon many things. It's their pH, their ionization potential, and then whatsoever drug dissolve can permeate through GI. And then from GI, it goes to the portal system, which takes to the liver, and whatsoever survives the liver comes to the blood. So you can see that in various steps, drug can break and not reach the blood, right? So here we are trying to see whether it dissolves fast, and we can see everything dissolves basically, which is good. Then we did the in vitro cell uptake study. It means if you take these formulations and keep next to the cells, like here we have used up cancer cells, U87 and GL261 cells, and see if they're kept next to each other in a petri dish, can these things can permeate the cell and go inside. Okay, this is simply a half job done. In vitro success doesn't show us that it will work in vivo. It's like if you are eating a sweet which is in front of your plate. It's very easy thing to do, but somebody has to bring that sweet to your plate, right? So even we have to see first it works or not. So we have used we put FITC, it's a fluorescence dye marker with amine dendrimer. You can see here, and you can see that it is crossing. The control has not much color here, green color, but you see it is crossing here. It means it's going into the cell. Similarly here, the C means carboxylic dendrimer. It also goes, but less than amine. You can see the color intensity here. But if you see the hybrid dendrimer, looks like a lot. You can see there's so many dots here. A lot of drug is going inside, like FITC going inside the cell because of the hybrid dendrimer. Same pattern is observed with the other cell. So we now know that dendrimer is helping drug to go, or FITC here to go inside the cell, crossing the cell membrane. And hybrid dendrimer is doing a better job. What we have done is that we have also, when you give something orally, so we know the dissolution is good, right? But what about permeation? So dissolution in the GI medium depends upon the solubility, which is a good thing. We proved by dissolution. Now the permeability is totally opposite of solubility. A drug which can easily dissolve may not permeate. A drug which could have permeated may not dissolve also, right? So what we have done that we have used this non-invasive in vivo imaging system. Here you can give them some fluorescent marker and put the mice inside of our rat inside, and we can do a live imaging of rat. We don't have to kill the rat. And you can see here that when we did that, and you can see that this is different dendrimers we tried here, but you see with hybrid dendrimer, this is an intestinal villi. You can see the green color. It means lot of drug is crossing from intestine. It indicates that lot of drug is going towards intestine and may cross intestine towards the blood. So this gives a very good indication that hybrid dendrimer is doing a good job even for permeation also. So what we have seen that when we use hybrid dendrimer, solubility is increasing, helping in dissolution, and not only that, it's helping in permeation to the GI membrane. So that way, it can go to the portal system, into the liver, and then from there, it can go into the blood. So we did that whole toxicity studies with Taxotere, the commercial formulation, and the hybrid dendrimer, and for the feed intake, hematological parameters, histopathology, and we found that our stuff is safer than Taxotere. This is histopathology of all the organs. I'm not going deep into that, but overall, thing is that the hybrid formulation is safer than the commercially available Taxotere formulation. So then we, the main thing, so we have shown by so many experiments that it should work, but is it working? So we gave it orally now, formulations to the animal, and then we measured the amount of drug in the blood with time. It's called pharmacokinetic. So you with different, at 0 time, you give oral gavage by oral formulation, like a syrup, to the animal here, and then at different time points, like half an hour, 1 hour, 2 hour, 4 hour, 6, 8, 12, 24 hours, we take the blood out and measure how much drug is there, right? If you see here, and also we compared it with the Taxotere IV formulation. So this thing you have to understand. Other formulations are oral, which is docetaxel we gave as a powder oral by putting in water, and there are different single dendrimers and hybrid dendrimers, and also we are given IV formulation. You can see that all oral formulation has a pattern. There is no drug and they increase and come down after 4 hours. So they all Taxotere given orally has the same pattern. And of them, the hybrid dendrimer, the blue one here, did the best job. And not only that, we compared it with the IV formulation of Taxotere also. So what you can see that the DTX, the pure drug given orally, we could not find any drug in the blood. Means nothing went into the blood. Amine dendrimer and all dendrimer formulation helped to increase the oral bioavailability, obviously. And of them, the hybrid dendrimer showed the best result. Not only best result, when compared the oral formulation of hybrid dendrimer with the injection formulation of Taxotere, giving IV directly into the blood, it almost showed the similar result. It means you see here, the AUC of hybrid dendrimer is 61 and Taxotere is 59. And on the contrary, hybrid dendrimer has a more MRT, means stays more time in the system. So what it shows? It shows that instead of giving injection of commercially available Taxotere, I can give you oral formulation and give you the same result. So you would say, why you will do that? Something is already in the market, why can't I use it? When you give something injectable, it creates problem because you have to go to doctor or the nurse, and that creates problem. It's called patient compliance. Instead of that, if you get something orally to take at home, you can take at your will. So first of all, it adds cost, it adds load to the system, and secondly, patient compliance. People don't go there. So if we can make something orally as effective as given IV injection, that would be very beneficial for the patient. So we have shown that these hybrid formulations are behaving as IV formulation, but given orally. So that was the first success of our objective one that we have shown that works. So that was the first part, to show that we can increase the oral bioavailability of a drug by entrapping in dendrimer. But the main objective was not that. Main objective was, can we create a multifunctional nanodevice? So what do you mean? Many of you hear about drug targeting, right? What is drug targeting? Drug targeting is based on the concept that in case of disease in certain area, some kind of demand changes. Like in case of brain tumor, brain needs a lot of iron. How will it get iron then? It overexpresses transferrin receptors. In case, sometimes they need more of folic acid. So they overexpresses folic acid receptor. That's how it works. So then from blood, when the folic acid comes, they take all the folic acid or all the transferrin. So basically, whenever they need something, and mainly many cancer and inflammatory conditions, there are certain receptors which overexpresses. So if we create a ligand for the receptor, what it will do? It will help you to take your drug into that area more. So when you give something, let's say by IV injection, what happens? It goes all over the body. So main problem with cancer medication is that it works in vitro if you keep next to a cell in a petri dish, but to reach to the cell, it has to go to all parts of the body. Obviously, these drugs are toxic. So in the process, they start killing other things in the body before they reach there. It's like if I have to reach the airport and I have a very big truck which has all the things coming outside, pointy thing coming outside, I may reach airport, but in the process, I'll be hitting many cars while going there and killing many people. So this is called side effects. That's how cancer drugs have so many side effects, and they create problem for the patients. How to avoid that? The main thing is that to make sure that your drug reaches where it's supposed to go. So for example, many of you, I hope you will use Google Map or GPS nowadays before you leave home. So you type or basically input your home address of where you have to go, and then it tells you where to go, right? And with nowadays unmanned car is coming. Many companies like Tesla are doing it, where the car, nobody will drive the car, and there's an address there. Car will drive you to your place without any help. Similar type of thing we are trying to create. And why we are doing? So what we are trying to do is that we will use a ligand which is specific to the receptor, which overexpresses in the particular disease condition. So what they will do is that it's like you create a delivery system, either liposomes or dendrimer or nanoparticle, entrap the drug and put that ligand into that. When you inject this thing, the ligand will help to target in that particular area. So till now, people were using one ligand like folic acid or transferrin or mannose and many other reasons for targeting. Different kinds of targeting depends upon where they will target. So I thought of this idea of using why one target, right? If you see a lock and key method, if you see your key, many key have many notches, not one notch, right? So what if we create a multiple notches, and there are chances that may have a better targeting efficiency. So here what I'm trying to do is that I'm trying to attach two ligands in a single system to check whether it increases its targeting efficiency or not. So, and we are trying to create an IV injection, not oral here. So for this research, we have took folic acid and mannose, folic acid and mannose. And here you see dendrimer is here. We attached folic acid here, one conjugate, and then we took the pegylate dendrimer in the other case and attached mannose. So now we have two constructs, one with dendrimer folic acid, one with dendrimer mannose. So this will be used as control. And we did all the analysis and found they work. Second approach, which I want to do, is having a multiple targeting ligand in your. Nowadays in India, people use double SIM, right? Why use double SIM in a single phone? Because you can get connection from all possible. If you have Reliance and I don't know what company, Jio or Airtel. So you can get calls from all companies basically, right? All possible companies. Similar thing I'm trying to create. In a single dendrimer, I'm creating two different ligands. So that when it goes to the brain area, it sticks very strongly, so it can go inside the brain. So we made single targeting ligand, folic acid and mannose. And here what I'm trying to do is that in a single dendrimer, I attached both folic acid and mannose. Now I have two antennas, not one antenna. In a single system, I have two antennas going like this, right? Folic acid and mannose. Otherwise, first I made dendrimer with one folic acid and one mannose. Okay, but making this thing is very complex. A lot of chemistry is involved. I have to attach folic acid and cover the whole thing, then attach something, then remove something. A lot of chemistry is involved, very complex. But we are using. For that, to come up with this, I came with the new concept. So this is how this, let me first, so thing which I name as a multi-construct. In a single dendrimer, you have folic acid and mannose. So we found that we can attach 6.6 folic acid and 14 mannose. But I came with a different option because of so much chemistry involved here. I thought this is good. So I have used a hybrid dendrimer concept to make it simple. So if you remember that I made a single ligand one with folic acid and with mannose, and then I used the click chemistry to click both of them. You see now here, so if you see the last multi-construct, in a single dendrimer, I am attaching two ligands here. In a single dendrimer, it's like this, a dendrimer, single dendrimer, and two ligands, two antennas. Here I have used two dendrimers, one has folic acid, one has mannose, and I clicked those two dendrimers using hybrid dendrimer. So now I have two dendrimers clicked together and each one have their own antennas. One has folic acid, one has mannose. Like now it's like this. If you see me here like this. Before, one dendrimer has two ligands like this. Now what I have taken that in one dendrimer, I put one ligand. In other dendrimer, I put other ligand and I click them like this. The two dendrimers having two ligands. Why? Because easy to make it. Very easy chemistry to make it. Otherwise, very difficult to make it. So now we will test it. So now we have dendrimer with one ligand, folic acid, and dendrimer with mannose. And then we have a nano construct where one dendrimer having two ligands. Now we have a hybrid dendrimer having two dendrimers, two ligands clicked together. Then we'll see what happens. And then we did TEM to see whether it works or not. You see the dendrimer alone, and this is dendrimer with folic acid, and dendrimer folic acid after the drug conjugation, which is docetaxel. Similarly, dendrimer mannose and dendrimer mannose with drug on that. This is multiple conjugate. You can see multiple conjugates bigger compared to these dendrimers here. So both in this dendrimer, folic acid and mannose both attached to single dendrimer. Okay, and after loading drug, it looks like this. And see H. It's very interesting thing here. H. Hybrid dendrimers. You see, it looks like a beads attached together. Remember, I told you I'm taking different dendrimers and I'm sticking them. You see, this is what's happening. This is dendrimers and they're stuck together. So it means it's working. Not only that, after loading this with drug, it looks like that. Now, what is my idea? So there are two things. One is targeting. What happens after targeting them? So there's a drug, you attach like a GPS, right? You reached airport where you have to go to address. What happens that? If your doors are totally closed, they're sealed properly, though you will reach there, you will not come out and not take your flight, right? So drug must release at the target area. The targeting ligand or GPS will make sure the most of the drug goes at that area. After reaching there, you have to make sure the drug releases there. So tumor has some physiological properties, not properties, has some changes happens, and that creates intracellular acidic pH. So tumor area has a low pH. So what I have done is that I've asked my student to take this thing and put in low pH, tumor pH. You know what happened? This stuff, when kept in low pH, you see, it breaks in two pieces. So basically it's cleaving the dendrimers and dumping all the drug. So what this will do now? That hybrid dendrimer will entrap a lot of drug. It has two targeting ligands, will keep move inside the body. Ligands will make sure that a lot of drug goes into the brain, and as soon as it reaches brain, it breaks and dumps all the drug, toxic drug, only in the brain, not anywhere else. And that we have shown with the real pictures taken through transmission electron microscopy. So then we did all the solubility studies, stability studies, and we found that all the formulations were very soluble and stable. Similarly, we did the biocompatibility profile. You can see Taxotere compared to Taxotere, all formulations were very same. So that's good. Then release, you see the Taxotere given because we are trying to give IV here. We want slow release. Taxotere market comes as a micelles basically. They had some micelles and some other stuff to make it a formulation because not water soluble. So all the drug came out very fast, but you see the Dendrimer formulation release the drug very slowly. So which is a good thing, because why you want the drug to release slowly? Because after you give injection, it takes some time to reach the brain area. You need some time to reach the brain area. And then if it keeps on moving, it's a more chance to reach, keeps on coming back, keeps on coming back. And more chance of sticking to the brain area and go inside. If all the drug releases, same time, it will excrete because they're small drugs through kidney and various reasons. Okay, so now we want to see that these formulations we made, like with targeting ligand, is this helping to go into the cell or not? So you can see here with GL glioblastoma cell, brain tumor cells here, control cell, nothing, literally nothing. When you attach Dendrimer alone with FITC, you can see some drug is coming inside, and with folic acid, some more drug. Mannose intensity is less, but it's more dense. Nano construct slightly better or same as single ligand, but you see hybrid Dendrimer, the intensity here. It's very intense here. It means hybrid Dendrimer with two ligands attached like this is helping more drug to cross the brain cells and go inside. Not only that, so if these ligands are helping, these cells, receptors are helping with ligand receptor interaction to go inside, can we confirm it? So what we have done is that because we are using FITC here and all those things, so we use the same receptor by the competition. So what we have done, if you're using folic acid, we first add a lot of folic acid, then we gave our stuff. And you see, not much is going inside. It means these receptors like folic acid or mannose are responsible for them going inside, because once you saturate the receptor with something else, these are not going inside. You see, nothing is here, nothing here, nothing is here. So it proves that folic acid and mannose are the main reason that the drugs are going inside. It proves that hybrid Dendrimer works best among the, and we proved with one more cell line here and one more cell different marker. All the time we got the same pattern. So then time to test on the real animals. You can see here, we used mice here, and we used a stereotactic surgery to give C57BL/6J mice with the glioblastoma. You can see the picture here, how it is done under the microscope. And the GBM cell line were injected directly into the brain area. And you can see how it looks. On the day 7, this is how the GBM looks. And day 14 looks like this when the mice dies, basically. Just to give you a visual idea how the brain tumor looks. See, day 7 start making like a simple road, and here it kind of spreads and start killing the brain. Again, for a better idea, you see day 2, how the tumor looks after injecting. Day 3, you see the day 4, day 5, day 7, day 10, and day 14. Just to you can see that how tumor mass increases. So before starting the real experiment, we have used we put some dye on dendrimer and injected into the mice. And as I told you that we have a machine where we can see live animal without killing it, and we can see that this is a brain area. This is a brain area. You see a lot of drug is growing into the brain area. So we can basically confirm that this formulation is helping a lot of drug to go into the brain area. So then we did the experiment where we have taken the GBM like a cancerous inoculated cell line inoculated mice, giving them formulation, and also we did the pharmacokinetic of them same way, but this time given IV, no oral. You can see that all behave same because the drug pattern is same after giving IV. Not much like hybrid dendrimer is slightly better than other, but not much difference among all the formulation when given IV. Hybrid is slightly better than even Taxotere, but overall they all look same. But when we did the tumor regulation study, means how tumor is controlled after giving these formulations to the rats, you can see that control the group where no drugs were given, only the GBM cell lines were incubated in all the animals, but here no drug is given. You see on day 14, this is like the tumor looks 100%. Okay, you see that when you give Taxotere, commercially available formulation for brain tumor, it has only 23% reduction. When we give Dendrimer docetaxel, which is Taxotere, entrapped in Dendrimer, it shows 19% reduction. It worked even not good even as Taxotere. But when you put folic acid on Dendrimer and then entrapped docetaxel, you see we got 48% reduction. With Manos single ligand, we got 52% reduction. Right? So what you have seen here, that Taxotere does better than control. Obviously, it's an anti-cancer drug, will show some effect. 23% reduction on 14th day. And when you attach a ligand like a folic acid, a Manos, they are doing better than Taxotere. Instead of 23%, 48% and 57% tumor area reduction. And then when you're using two ligands in a single dendrimer, you see 64% reduction, better than single ligand. But even among dual ligand, the hybrid dendrimer concept shows 71% reduction, which is a very big reduction basically. So what we have seen is that, just to summarize everything, everything here, the Taxotere works better than the control. Dendrimer with one ligand works better than Taxotere. But Dendrimer with two ligands works better than Dendrimer with one ligand. That proves my theory that instead of one targeting ligand, if I use two targeting ligand, that will show better results. But among dual ligand also, the hybrid Dendrimer which I developed is better than the construct idea where we have two ligands in one dendrimer. And the reason could be that if you're moving like this, if you're very much next to each other, it's difficult to have interaction compared to the proper distance between those two ligands. So they can stick nicely, right? So spatial difference is also very important. So what I'm doing with hybrid dendrimer, I'm providing a lot of spatial difference for them to interact with the receptors. So then we did all the immunohistochemical analysis with tubulin, VEGF, and other COX and all kind of markers, and we found a pattern. I'm not going deep into that. Here is a survival plot, means how many days the mice lives, right, after during experiment. So we tried two approaches. One is preventive and one is therapeutic. Preventive means preventive will give from day one, every day we're injecting all formulation while we are transplanting GBM cell line into the brain, right? So when you transplant your cell line, GBM, cancer cell line into the brain, drug is already there, preventive. Therapeutic is after 7 days, when the brain tumor is developed, then we start giving the medication. So we tried both approaches, therapeutic and preventive. So let's see therapeutic first, the lower one first, right? So basically after you see the 7th day, after the brain tumor is formed nicely, we start giving formulation every day. You see the normal rat, which is not given anything, dies around day 12 or 13. Sorry, mice dies around day 12 or 13. Taxotere dies around 18 days, so increases the lifespan. And if you see that Dendrimer folic acid, 30 to 33 days, and then mannose further better. So attaching a single ligand helps to increase the lifespan. Attaching two ligands also further increase the lifespan. You see the nano construct has around 39 days lifespan, but with hybrid dendrimer, it's like a 45 days lifespan. So you can see that compared to Taxotere, if I use hybrid dendrimer concept, I can increase the life of animal more than 2 times. So like in current time, if patient stays alive for 2 years, he will stay alive for 4 years, right? So that's what we saw in therapeutic mode. But we also tested what if we start giving medication before we injected the tumor cell line. Here you see that the normal control lives around 13-14 days, almost same to before, not much changes. But in case of Taxotere, living more, 22 days approximately. And same, the single ligand formulation, Dendrimer, increase the lifespan. And of the dual ligand, again, both doing better than single ligand. But you see the hybrid Dendrimer, 60 days almost, 59, 60 days. It increases the lifespan. So what it shows, that hybrid Dendrimer formulation is increasing the lifespan of these in case of brain tumor, and it's doing a lot better than commercially available Taxotere formulation. And we have shown by animal study, and we have shown by all the mechanistic study and histopathology, all those things. And we are so apart from that, we did some kind of safety studies for the body weight. They were all good and safe. So basically, we reported that by using the hybrid Dendrimer concept and dual targeting ligand, we can have 2 or 3 times better effect than commercially available Taxotere formulation. So basically, this is our kind of a summary of the work to explain to you. And one of the paper published in Nanomedicine, other paper we are still kind of writing. So yeah. So this is about the work. I think my time is almost, how much time I have? Hello? So I think, yeah, this is about my work. So I will answer any questions if you have.
From audience, there are 4 questions.
Yeah, sure.
I am reading that questions.
Can I see
From
them?
Nilesh Mahajan, he asked what kind of chemical interactions are there in hybrid dendrimers?
So there's no chemical interaction in a hybrid dendrimer. That's the reason I created it. So otherwise, to take two dendrimers and make a megamer needs chemistry. Here, it's a physical interaction. And I cannot tell you the recipe because it's my secret. I have a patent on that. But there's no chemistry involved in that. That's the reason. It's like taking two things and mixing it together. And they click to each other. Like click chemistry. You may have played with these toys, right? Kids play that, they take it together like Legos. You click it together and build the homes. Something like that.
Okay, hope you satisfied with the answer. Nilesh Mahajan ji. Another question from also Nilesh Mahajan. Tamam dendrimers are known to have cytotoxicity. How did you overcome it?
So this is a kind of a misconception about PAMAM. When you say PAMAM, it has many surfaces. So PAMAM dendrimer with amine surface has toxicity, but at very high concentration, right? So you're right in a sense that it has some toxicity. And I published a paper many years back showing the all toxicity parameters. At low concentrations, they are not toxic. Second, we are modifying it, right? So when you use PAMAM dendrimer with amine surface, they are more toxic. If in your language, I explain it. But if you change the surface to carboxylate or PEG or pyridine, they're not toxic at all, right? And here we are modifying the surface also here with PEG or folic acid, so they are less toxic. Second thing, we are using a very small amount here. We don't need large concentrations here. So they will not be toxic, and we have done the whole toxicity study here. Is that answers Nilesh's question? He can counter. He can ask me more question if not. If he has more question.
Okay, one more question from Manish Kushwaha. Sir, when dendrimers cross blood-brain barrier, it alters brain activity or not?
I don't know the answer as of now. This is a very good question.
Yeah.
It's a question that I need to answer by science, saying that what happens to the cognitive function inside the brain. My understanding is that because dendrimers are very small, they will excrete after some time or degrade inside. But it's a very good question. I don't have answer for now. That would be matter of my further research. In a few years, I will be able to answer that also.
There is one more question from Dr. Srinath Bal Kundi. He's asking, dear sir, can we deliver vaccine by dendrimers to immune cells?
Technically, yes. Yes, you can deliver. But depends upon what size you're doing it. You have to understand dendrimers which we use are like 14, 15, 20, 30, 40,000 molecular weight range, like a Dalton. So what size vaccine you're doing? So if you're using vaccine, don't use a full protein, use a part of a protein. Then yes, you can do it. And there are a lot of publications. If you just search online dendrimer vaccine, people have done it. But yes, it's possible. Other thing is that dendrimer also helps in stabilizing it. For example, DNA or RNA, if you just inject DNA, RNA in the cell, the polymerase will come and click it. But when you trap in dendrimer, it cannot clip it.
There is one more question from Megha Chouhan. She is asking about, can we use plant chemical constituents to form dendrimers?
So dendrimer is an architecture in a sense that, let me go back and explain you. Oh, let me explain you, go back. Sorry, I have to go to the first page. Okay, so here's dendrimer, right? It's an architecture. Architecture where, let me see if I can draw something here. Bear with me, okay? Pen. Okay, dendrimer is an architecture. How is formed? If you need a 3, I'm drawing something. If you can, sorry, I'm not using mouse, so it will not be very good. but just explain. It needs a 3-armed structure first, like Trishul, right? And then you keep on adding other chemical to start the next layer. So you keep on adding like this. So one more layer, okay? Here, this is a generation 1 dendrimer. Then you keep one more layer. You have to keep on adding something which has 3 branches always. If you use 2 branches, then it will be like a straight line either way, right? You cannot create a 3-dimensional structure. So then here, you keep on adding again 3 structure where the first part sticks here, other 2 parts grow out. And you keep on doing that. After some time, you see it start taking the shape of dendrimer. Okay. So when you're asking about using what was her question about
plant chemicals? Plant chemicals. Yeah.
So plant chemicals are not very simple chemicals. Like here we are using amines and acrylic.
It's a mixture of various chemical constituents.
It's a mixture of various things, right? So you cannot. So my answer is answer to your question is no, because they are not a pure chemical. They are like, you know, all complex glycosides and all the alkaloids and all those things. Not easy to do chemistry over that. But you can use plant chemicals to entrap in dendrimer. So just to tell you that one of my patent when I was in industry was purchased by a company called Agarium in Australia. They are using it for deliver for agrochemical. It will be marketed soon.
Sir, one thing I would like to ask you in this question, which kind of chemical constituents we can say to approach via dendrimers for the novel pharma?
To entrap it to make it.
Okay. And there are chemical constituents nature like alkaloids, glycosides.
You can do anything. It all depends upon the size. It's almost like a cage, right? So what you can fit, right? So any small molecule you can entrap in dendrimer. Like very much it goes here. Let's say inside. Either a molecule can entrap here like this, if you see the picture here, or you can do chemical conjugation here. So let's say, let me change the color, if I can change the color. Ink color. Now I want to touch folic acid. I can do chemistry, attach folic acid here. I can attach mannose here, for example. And you want to attach something else, you can attach something else here. So you can do both physical entrapment and you can do chemical conjugation, right? So if you want to, let's say, for example, the thing which I have a patent, many more apart from me in that project, where we have shown that, for example, many cases of root disease, if plant root has some problem, if you spray medication, what happens? Most of the medication is stuck where? In the soil before reaching to the root, right? What soil is? Soil is silica. What is silica charge? Positive charge.
Okay.
If you make sure if you entrap a drug, a pesticide in the dendrimer, which is negative charge, positive charge, what will happen? It will not interact with silica and go deep into the soil and help the drug to reach the root.
Okay, thank
right?
you, thank you.
So these kinds of things can be done.
Okay. There is one more question from Anchal Parnami.
Mm-hmm.
He is asking, do we need to purify dendrimers?
Yeah. So making dendrimer is not easy. That's the kind of disadvantage with dendrimer compared to liposome. So I tell you example. So making liposome on nanoparticles or micelle is very easy. You make in situ means you take lipids or monomers or any kind of surfactant and mix it together, and after certain condition they form liposome, like critical CMC or micelle, something like that. They prepared in situ, and you add drug while they are making it, right? So this is called their drug encapsulate into that. Drug is inside those things. Dendrimers are not like that. Dendrimers are like you first make the instrument, you first make the bowl by chemistry, and then you add drug over it. So drug is not adding inside to here. So making dendrimer is not easy. It's a chemistry thing. Until unless you're a good chemist, you cannot make dendrimer. You have to work with some chemist or buy it. Like Dr. Tamalia sells dendrimer, nanosynthons, and Dr. Pawar can help you. I can give him the information. You can buy it from here. There it's a lot easier. You can buy a small one and work with that. But not easy. Making dendrimer is not easy.
There's one more question from Sumit Sahu. He wants to ask, explain methods are used dendrimers synthesized.
Pardon, explain?
Explain methods.
Of making dendrimers.
Yeah, used dendrimer synthesized.
So making dendrimer is not easy.
Okay.
I'm telling you, I cannot explain here. You can read online, there are many papers here. Basically, it's amine and acrylate chemistry. You take an amine dendrimer like either ammonia or ethylene diamine. It must have more than 3 branches. I explained you to have a 3D structure, right? And then you attach acrylate over it. Then you attach again ethylene diamine over that. So one of like ethylene diamine has CH2, CH2, and H2. It has 4 branches. So one branch will stick here and 3 branches will open outside. Then you attach acrylate. Each branch will have their own acrylate. Then again attach amine. It will open 3 again. So it keeps on adding. It's a simple mechanism. Regarding chemistry, yeah, until unless you have good facilities, you cannot make it.
Okay. There is a question from Harshit Sharma. Harshit Sharma want to ask, is there any adverse effect of dendrimers in body? Yeah,
it is. There are adverse effects. Like the one instrument, I think Nilesh, if I am not wrong, he asked about the cytotoxicity. So dendrimers which have positive charges are slightly cytotoxic, right? But then you can modify those dendrimers, use PAG or something else, or when you entrap a drug that reduces its toxicity. So yeah, it has some issues, no doubt. Everything has issues. Means everything which you add in a body is toxic. Even if you drink a lot of water, you will die.
But
the thing is that what concentration you are adding into that.
It may depends upon the concentration, maybe.
Yeah, something depends upon the concentration. But yeah, it has toxicity issues. Other thing is that, like one student asked the question of effect on brain function, right? What happens when dendrimer takes drug into the brain? That's good job. But if dendrimer stays there, is it affecting the brain in some other way also, right? So these are very unknown things which we need to figure out.
There is one question from Dr. Ram Narayan Prajapati from Jhansi, Bundelkhand University. He's asking, what is the cost of PAMAM dendrimers and from where we can purchase, as you told earlier, I think.
So Dr. Prajapati, you can buy from nanosynthons. Dr. Tamalia, who is inventor of dendrimer, they sell it. And if you need help, I can help you. I'm a consultant with this company and I can help you. Ask Dr. Pawar, he'll give you my email or my classmate. Do online without my help also. Company name is Nanosynthons, Nanosynthons, Nanosynthons.
Mr. Professor Ashish Singh, I want to ask, can dendrimers be explored for Alzheimer conditions?
Yeah. Actually, I got some email. So like 4 or 5 years back, I published a paper where I've shown that I can use nose to deliver drug into the brain directly. So I received communication from one big Alzheimer scientist. They're interested that if I can do that, can I help in Alzheimer drug also?
Okay. There is Mr. Kamlesh Kale want to ask, are there any chemical changes in brain after drug interaction?
So after drug interaction, there must be some changes, means obviously, but these drugs are there in market for a long time. And obviously it needs to be studied. Docetaxel is not a new drug. It's a very old drug from long time. Lot of literature is there. But I think in case of brain tumor, when person is dying in few months here and there, increasing lifespan is more important than affecting brain here and there. But yes, it may affect.
Dr. Sunil Prajapati wants to ask how you plan the animal study.
So basically, we are trying for the brain tumor. So the whole idea is that in that second part of the thing, the whole idea is that testing one ligand versus two ligand theory, which theory I developed, where I can say having two ligands, like two notches in a key, will do a better job than one notch in a key. So basically it was easy that we decided on the brain tumor, the cell lines we arranged, and we thought of two models, preventive model and therapeutic model. Therapeutic model is a realistic model where you start giving medication after somebody has brain tumor. So we gave medication after tumor has developed. In preventive model, we start giving before, thinking that what happens if we give it before. And we have shown that if you have an idea, somebody is prone to brain tumor and give that medication before, it will delay the brain tumor. So, and then basically we injected the brain tumor into the brain directly, the machines. And every day we gave formulations and took the blood at different days. And basically the day 14, and then basically the day rat dies, we cut the rat or mice basically and remove the brain and look for histopathology and all other things.
Okay.
And remove all the brain, various tissue organ also to measure the amount of drug in each organ. So
there is Professor Mahen Singh Asawat want to ask, is any dendritic chemical composition can be possible which can make precipitate with the proteins?
So dendrimers are very water soluble.
Yeah.
So dendrimer, when you make, these are covalent structure. So difference between micelles and liposomes and all those things, they can break inside the system. Dendrimer will not break. It's a covalent structure, right? So I don't think it will precipitate. It can interact with protein. For example, it will interact with albumin and all those things. Yes, there's a possibility is there, but precipitation will not happen. They are very highly water soluble. Actually, I was the first one to use dendrimer to increase solubility of drug.
Okay.
So they will not precipitate as such. They can interact.
Also, yeah. He's also against, wants to ask his possibilities to incorporate anti-tyrokinase enzyme in dendrimers for dermal formulation.
Miss, yeah, you can do that. So you want to use that for dermal, transdermal, right?
Yeah, yeah.
So yeah, I was the first one to publish dendrimer for transdermal. I've shown dendrimer can be used for transdermal. Only issue with the enzyme is their bigger structure, but you have to see. I've used small molecules, so crossing through skin with bigger structure could be an issue, but you can try. That would be a big, first of all, enzyme is bigger structure. Entrapping dendrimer would be a problem. Second, crossing skin also would be a problem. So not impossible, but you can just try, entrap it and take any skin of rat and check the permeation to the French diffusion cell.
And the last question from the Deepak Srivastava, can virtual model produce more relevant results?
So I don't know the question. What virtual model?
I don't know also what you want to do.
So I think so this is not a model, right? This is a real. This is not a molecular modeling. It's a real research. Okay. Things were made, injected in the brain tumor cases, and we have shown the results. Like it's like a typical animal study before going to human trials. So I'm not sure he can clarify his question. I'm sorry, I'm not able to get the question.
So thank you so much, sir.
Modeling will never be an alternative for the real work because virtual modeling depends on certain conditions right? It's a mathematical model. It gives you an indication, right? But not, it's a good tool to support your data, but it will never kind of be alternative for real animal study.
Okay, thank you, sir. Thank you, sir. Nice presentation, nice information session, and nice question answer session also. Now I would like to request to Sri Shyam Rathore sir to say a few words about the session, and to our honorable speaker, Dr. Chouhan.
Yeah, I already said in the starting that it's very good, and may with your all these efforts, we will get a very corona-free world or planet very soon, with the efforts of all the pharmacists and everyone there. And thanks for giving us the time and opportunity to interact.
No, thanks for inviting me. Thank you.
Yeah.
And the last, I would like to inform all the participants, tomorrow has also a session on IPR, and along with the session, we are going to circulate a link for feedback of the today's session and tomorrow session. So all are present tomorrow and participates in the feedback, as well as same time we are going to circulate link for the certificate also. So thanks to all of you for listening very carefully to us. And thanks to Dr. Chouhan for giving us your valuable time, and very much fortunate to with us, you all to with us. Thank you, sir.
Thank you. Thanks for inviting me.
Yes,
Thanks,
sir.
Mr. Paul.
If you want to say something to us,
nothing, Miss. I will only say one thing. If anybody who has students here and all those things, that when you're doing research, be sincere. I'm sorry to say this thing, and sometimes people don't like it, but I will still say it. Don't manipulate the data. Okay, it never helps. Okay,
it's a if you manipulate the data, it's kind of a scientific misconduct.
I don't care about even misconduct also.
Yeah.
Means you're wasting chemicals, don't work. If your boss allows you, don't do anything, write your thesis. I don't give a damn about it. Don't waste chemicals. If you really do, there are so many things to solve. Everybody doesn't have to make a very hi-fi thing. Solve simple issues, solve tablet issues, capsule issues, work what you have. Okay? By manipulating data, you're killing yourself, you're killing your scientific spirit. Wrong data never helps, because nothing will come out of it.
Yes, sir.
Right? And don't think you don't have a caliber.
They misguided also.
Basis. Like I came from Sagar with all of you guys, not like anybody comes from anybody else, right? So we all can do that. That's not important. Being honest. And I tell this thing, many people, I'm telling you again. I was doing my PhD, I was registered in Sagar, but I worked at Indian Institute of Chemical Technology, Hyderabad. 4 years of my PhD, I didn't have a single paper.
Yeah.
4 years of PhD. I didn't have a single paper. And all my friends here and there, juniors, have 10 paper, 12 paper. And I was thinking, oh, what I'm doing wrong. But you know, I published just one paper. And that brought me to US without any help. Donald Tomalia, I went to a conference in Germany, and he saw my work. He offered me a job at the conference when I was a student.
Yes, sir.
The guy who invented Dendrimer, whose name is for Nobel Prize as of now. So what I'm trying to say is, but I never manipulated. I believed. I invented an anti-inflammatory property while doing a PhD. And many people said to me, my friends, oh, manipulate, it's not possible. I said, no, this is what I'm seeing. I believe what is I'm seeing. Then I changed to get published here and there. I didn't believe, then I repeated. And I found a new property.
Yes, sir.
So you all see, stop listening to people who say manipulate and all those things. It never help you anything. If you don't have facility, don't solve big problems, solve small problems. There are many problems. Yes. But if you're honest with your research, it will take you to big places. If you're not honest with your research, it will never help you. Once you start manipulating, you will never learn. You will always manipulate. And then your students will manipulate. So don't take me the wrong way. I'm not saying, I'm not saying the wrong way, but I'm just telling you very frankly that we have to think about that how much, when you do research, there are two things possible. One, are you adding something in academic, like coming with the concepts and all those things, or
Yes,
you're bringing something in market, right?
yes,
And it has a lot of commercial. If you invent something, for example, coronavirus is coming now, right? Every company is behind to make a vaccine or diagnostic kit for treatment for coronavirus.
yes,
If you are working on, like somebody asked me a question about molecular modeling. If you are working on molecular modeling, fine, take all the drugs and basically match with coronavirus if you get some model basically and see which one sticks basically. People will be behind you to do this kind of work. So do the work which is relevance, right? If you understand that way, that if you have a shop of samosa or kachori, you're not making a real one. No one will come to eat eat your stuff, right?
yeah,
You have to make a real samosa and real kachori or real pulki, whatever it is, right? So believe yourself. Don't do science because somebody else is doing. See, I never publish so many work. Once I do work, I don't change the drugs and publish something else because it doesn't excite me. In the same work by changing drugs doesn't help by any means. There should be clear reason why are you doing it. When you do your work, it should excite you. That okay, if this is successful, I will get this. Outcome is clear out of it, right? So make sure, and you don't have to work on creating robots, or I didn't create robot when I was doing master's degree.
okay,
I'm doing now because I have facility, I have infrastructure,
yeah,
I have understanding. Takes time, but try to solve simple problems. Don't try to solve complex problem if you cannot solve it. If you don't have resources to solve it. If you do that, just think about that. India, we have so many PhDs come outside every year.
yes, sir.
How many of them are helpful? How many masters are helpful?
Yes, sir.
How they are contributing? So we have to be critique ourselves and see how best we can help the scientific society.
Yes, sir.
That's all I have to say.
Thank
And
you, sir. Thank
stay
you.
inside your home. Don't go outside.
Yeah. Thank you, sir, to share your views on research aptitude, what kind of research we should have to follow, and what should be the science culture. Actually, science culture is very unmanipulated. It should be not manipulated. So thank you, sir. Thank
Do a
you. And
simple thing.
last,
Do a
yeah,
simple
yeah.
thing. Yeah.
And last, I would like to thank our BOG member and chairman, Sri Shyam Rathore, sir. He is always promoting us to do newer things, and always stand with us to do the things which is benefited to the society. And I'm very much thankful to Shyam sir and other BOG members and my dear participants who makes this session very lively. So thank you all. We will meet again tomorrow at the evening time at 6 p.m. with the various kind of applications. The theme of tomorrow session is applications on IPR, intellectual property right, with Jaideep Goswami. So we'll meet again tomorrow. Thank you, sir. Now I am going to end the meeting. Okay, sir. Thank you.
Bye, guys.
Okay.